Okay, good morning. Good morning. Hello. Okay, so I don't know if it's that I didn't take my allergy medication or I have no idea, but God, things are crazy this morning up here. Okay. We're in molecular biology. If you haven't figured that out this semester, there's some major trouble coming. Okay. Pick up your exams and recitation, those who haven't. The gene expression topics need to be finalized this week. More than anything for the sake of your sanity. Because if you keep on wishy-washying around, then you're going to have no time to do the actual project. Picking the topics is just step one. Did I see your hand? No. Okay. Remember, this is a proposal, not a report. A lot of people are choosing things, and they're just like, well, it's expressed really high, and it's part of the immune response, so that makes sense. And I'm like, yeah, so what are you proposing? So ask yourself, as you choose a topic, what am I proposing? I'm asking someone for money to study something. If it's something that there's nothing to be studied because it's well characterized, especially throughout your virus, then no one's going to give you money, right? So you need to be proposing a project. Do you need to know exact experiments? No, you need to know how to ask scientific questions. Like, does this affect this? Does this bind to this? These kind of things. Okay, exam 3 is coming up. We won't worry about that. Okay, so far, lots of stuff. We've been talking a lot about transcription, and over the last few weeks, we've talked about differences between DNA and RNA, and differences between DNA polymerase and RNA polymerase. We studied the transcription cycle in bacteria, transcription initiation, we call too, in eukaryotic cells. We understand the concept that transcription is coupled to RNA processing. We learned about the capping reaction, splicing, polyadenylation, and we learned those mechanisms. Okay? So now we're going to move on. Are we going to move on? Yes. So next, the RNA goes out of the nucleus and it gets translated. But before we move on to any talk about translation, we're going to talk about regulation of gene expression. Okay? So big picture, right? This is the path of RNA, right? So you have DNA, RNA, so transcription, you get RNA, you get processing, you get mRNA, it goes out of the nucleus, binds to the ribosome, hopefully becomes protein, might be modified, and becomes some active protein. And it does something, right? That's what's going on here in the big picture. But every step of the way is a possible point of regulation, a possible point to interfere for some interference or some activation or something to happen to either turn something on or to turn something off, right? You can have transcriptional control, which is really what we're going to be talking about today. What activates gene expression? What allows transcription to be turned on or keeps it off? That's your transcriptional control. You can have RNA processing control. We talked about that, right? A good example is alternative splicing. There's a lot more of them. There's RNA editing. Also, there's you can decap or remove poly-A tail to destabilize an RNA. All these things are going to prevent your RNA from leaving. Well, not alternative splicing, but a lot of things that mess with the appropriate look of an mRNA, cap, tail, no introns, are going to prevent an RNA from getting out of the nucleus, so it'll never be translated, it'll end up being degraded. If it does make it out, there are steps of regulation based on transport and localization. So that's, whether RNA can get out and where it goes in the cell, because RNAs are going to go close to where they the resulting protein needs to function. Then you can have another point of regulation. You can have microRNAs or siRNAs either prevent translation or target the RNA for degradation. So you'll have inactive or degraded RNA, and then you'll have no protein. That's a way to shut something off. You can have microRNAs that are going to prevent translation. You can have other mechanisms that might prevent translation, so no protein. If you get successfully past that and you have protein, you can still regulate at another level, right, through regulating protein activity, through phosphorylation or some kind of post-translation modification or degradation. Tons of mechanisms for regulating gene expression, right? And if you think about it using this kind of picture, and you picture in your head DNA to RNA, all the processes that need to happen to get to a functional protein, think of it not as a list of things you need to memorize, but as a process that needs to happen to result in a functional protein. Then it'll be easy to think through each step and say, okay, what's next? Processing. Oh, right, processing. There's a bunch of regulation out of processing. I have my mRNA. What's next? It needs to get out of the nucleus. Oh, there's another step. So if you think through it like that, it's not a lot to remember. It just kind of makes sense. For those who like lists, so here are some general mechanisms of transcriptional regulation. And this is mostly, let's see, this is mostly what we're going to talk about. The first two steps are mostly what we're going to talk about over this week. It all depends on whether or not, because this is transcriptional regulation as in, does transcription happen or not? And so a lot of that revolves around whether or not RNA polymerase can bind to the DNA. If there's a repressor there that prevents it from binding, or if an activator is required and it's not there, you're not going to have gene expression, right? You don't have RNA polymerase binding to the promoter, no gene expression. Gene is off. Activators and repressors, they play a huge role in this. You can regulate initiation. You can regulate the ability of RNA pol to get past that abortive initiation into productive RNA synthesis. You can regulate elongation and termination. Sometimes the RNA itself can regulate its own transcription by forming secondary structures. So you can imagine if RNA polymerase is moving along and there's some sort of secondary structure forming that maybe will slow down transcription. And then there are the mechanisms of post-transcriptional, different from post-translational. So remember, post-transcriptional means after the transcription. Mechanisms of post-transcriptional regulation that I just talked about based on that figure I was showing you. Regulation of RNA processing, regulation of mRNA export and localization, regulation of mRNA translation, selective destabilization or degradation of mRNAs. And then, of course, my favorite, regulation of protein activity through post-translational modifications and targeted degradation or inactivation and relocalization. So what's an example that we've learned in this class of how protein activity is regulated through post-translational modification? We've already learned a million examples of that, right? Who wants to give me an example of that we learned, I don't know, in the first few weeks of class? Yeah. Acetylation and methylation. And ubiquitination, that's awesome. Right, remember the histones and their activity is affected by post-translational modifications because whether or not they're acetylated or methylated or whatever regulates whether or not different proteins will be recruited and whether or not histones will be moved around or bind tightly to DNA or whatever. Right, so that's an example. And this same concept goes all the way through some molecular biology. If you have a protein that's being made and you ubiquitinate it, sometimes monoubiquitination will just send it to another part of the cell, or SUMO or phosphorylation will allow different proteins to bind to each other. So there are a lot of these modifications that happen that regulate the function and the activity of the protein. Okay? Questions? Hopefully you guys will all leave this class completely understanding that concept. Okay, so let's have a little conversation. So we've got the same genome and different RNAs and proteins, though, right? Same genome in every single cell type, but different RNAs and proteins. So that's what we're talking about. I'm trying to bring it back to the big picture because some people have been visiting me and saying they're completely lost and drowning. And I want to make sure we can keep on going back to the big picture so people understand where all these pieces are fitting together. Right, so you've got the same genome, same DNA, same chromosomes. Let's talk about eukaryotic cells for now, but we've got different RNAs and proteins, right? So different cell types, they're synthesizing different sets of proteins, right? Does that make sense? Of course, right? A red blood cell is going to need different proteins than a neuron, right? Maybe. Okay, so who can tell me what kinds of proteins might be common to all cells, and therefore it's likely that transcription might be constitutively activated? Yeah. Proteins involved in maintenance and generation of cell membrane. Okay. You don't have to be quite that specific, but yes, that would be one. What else? RNA polymerase. There's a great one. Yeah. So proteins that are associated with the normal functioning of a cell, that are not specific to the job of that cell, right? There's normal functioning proteins, and then there are job specific. Like, what's the job of a liver cell compared to a neuron, compared to a red blood cell, compared to a pancreatic cell? They all have different jobs, right? They all do different things. So they're all going to express different genes. So proteins there are some proteins that are only expressed in certain cells. You can think of the most obvious example of a protein that's only going to be expressed in one kind of cell. There's like one cell that's highly abundant in our bodies and super important. Really important, yes. Red blood cells. What kind of protein are they going to be likely to synthesize? Huh? Hemoglobin, right? Right. Do our neurons need to make lots of hemoglobin? No, that would be weird, right? So hemoglobin. So you could expect that hemoglobin is going to be, or globin genes are going to be regulated differently in all these different cell types, right? So that's where we're at. Okay, that's the kind of thinking I want you to be thinking at as we start to dive into all this gene expression and regulation of gene expression. Always say to yourself, whoa, what are we talking about? Oh, right, major concept. So there's variation. There's not only is this gene on or off, there's also variation in levels of expression of different genes in different cells. Maybe in one cell you'll have very high expression of some gene, and in another cell it'll be lower. An example that we've talked about is telomerase, right? Sometimes it'll be expressed very low, sometimes it'll be expressed high. If you're a cancer cell, it might be expressed really, really high, right? Okay, so another concept I wanted to bring up is that RNA does not represent the final product of gene expression. What I mean is there's no direct correlation between increased expression of RNA and increased expression of protein. You might think there is, but there's many places for things to go wrong after you get mRNA, or things to change, okay? Right? Now, it is frequently used as a measure of gene expression and a reliable measure, but I don't want you guys to think that is it, okay? You can have a lot of things that happen that are going to affect protein function. And so it's not, right, so you can have what did we see on that gene expression project? Sometimes like 3,000-fold increase in some expression of some gene. Does that mean there's going to be 3,000-fold increase in that protein? I don't know, maybe, yes, no. If there's a 1-to-1 translation ratio and nothing happens and it sticks around a long time, yes. But don't forget that you can have that RNA go out of the cell and be degraded, right? There are mechanisms to remove RNA before it ever gets to the ribosome. There are mechanisms to slow down translation of RNAs. There are mechanisms to degrade proteins. So just remember, it's not just lots of RNA, lots of protein, that's what it does. There's a lot going on. Okay? Any thoughts? I'm talking a lot today, I'm sorry. Please stop that. Take a break. Oh, by the way, I'm sorry about the faux pas with the quiz last night for those who are last-minute quiz takers. Hopefully you guys got to recoup after that confusion. Yeah. The quiz closed 2 hours earlier, so when people were like, ah! So hopefully you're supposed to all be done by like Saturday morning. So excited when they post the quiz that you do it all night, Friday night. Maybe you sleep the rest of the weekend to recover, right? Okay. Okay, so transcriptional regulation. What does this mean? I did get a comment from someone that sometimes the language I use in class is hard to understand. And I don't mean as in English is hard to understand. I mean just the terminology can be fairly heavy on the science and sometimes people just aren't used to it. So what does transcriptional regulation mean? That's someone who we don't hear from very often. How about Chewy? What does transcriptional regulation mean to you? What does it mean to you? I know, you can talk to a friend. Feel free to recover from your name being called out by the professor and talk to your friends. It doesn't have to be perfect. Huh? Shout it out. Pretend you're in an acting class. Upregulation of genes or cofactors involved in processing. Is that what I heard? Upregulation of genes is a result of transcriptional regulation. But let's try again. What does transcriptional regulation mean, Doris? That's good. Anything that affects whether or not transcription is going to happen. Anything that can activate or repress transcription. Yeah, so when you see transcriptional regulation, what we're talking about is activation or repression of gene expression. Are you going to get RNA or not? Okay? And I always want you to keep in mind, any step in the transcription cycle can be regulated. From initiation is what we're going to focus on the most, but all the way through to termination. There can be different points where things can be slowed down. Okay, so let's start by talking about regulatory regions. So these are DNA elements, so sequences of DNA that are semi-close to the promoter, usually. Sometimes quite a bit far from the promoter, depending on whether or not you're talking about bacteria or eukaryotic cells, but they're regions in the DNA that bind to gene regulatory proteins. I got an email this weekend about the quiz. Asking if gene regulatory proteins are the same as transcriptional regulators. So they are. They're the same thing. Depends on the textbook you read. But if you think about it, it's describing a function. Gene regulatory proteins are proteins that regulate genes. Transcriptional regulators are proteins that regulate transcription. Okay? And so they're really just proteins that are going to bind to DNA in some way and either activate transcription by helping to recruit RNA polymerase or repress transcription by acting to not recruit, acting to prevent recruitment of RNA polymerase. Question?
What's the difference between promoter and
So Tata box is part of the promoter. The question is, what's the difference between a promoter and a Tata box? Tata box is part of the eukaryotic promoter. There is a TA-rich region in the bacterial promoter, yes, but Tata box usually is referring to the a region within the eukaryotic promoter. So you could say promoter refers to any part of the gene directly upstream, downstream, that way, to the minus. I always get confused by upstream and downstream. From the start of transcription. So 10 to, I don't know, well here there's a region at minus 10 and a region at minus 35 on bacteria, but in eukaryotic cells it can be hundreds of base pairs before the transcription start site. So promoter is that region, and that's the part that's going to recruit RNA polymerase. Tata box just refers to one small sequence within that region.
I thought RNA polymerase was recruited to the Tata box.
By Tata binding protein? Yep, exactly.
Okay.
So the
The
promoter,
promoter is a larger region.
yeah, so this is a really great small thing that will clarify things for a lot of people. The promoter is a large region of control. You can think of it, it's the region of DNA that promotes transcription. It contains different sequences that bind to different transcription factors, but the Tata box is what recruits the Tata binding protein, part of TF2D, the first general transcription factor in eukaryotic transcription. So just think of it, it's a TA sequence within the promoter that recruits the first general transcription factor that then results in recruitment of RNA polymerase. Okay? I know there's a lot of new terminology sometimes, and it's hard to understand what is what. So whenever you find yourself in that, like, wait a minute, how do these relate? Please ask, because it'll be helpful to everyone. Okay? So when we're talking about bacteria, it's very simple, right? It's really about whether or not sigma factor can bring, which they're actually not showing in this picture, whether or not sigma factor can bring in RNA polymerase to the promoter, and then transcription can start. And so here it's showing a very simple system where you have a repressor, it's the trip operon, which we'll get to in more detail later, but where there's a repressor that will bind in this region right here within the promoter. They're calling it the operator region. That's pretty universal. It's usually a region that controls whether or not RNA polymerase can bind to bacteria. And the repressor, under certain conditions, will bind. And look, can RNA polymerase bind? No, there's a repressor in the way. So RNA polymerase doesn't bind, genes are off. Okay? Yes?
So all the genes in that sequence won't be expressed because RNA polymerase can't bind because of that small repressor.
All the genes, well, depending on how many genes are controlled by that operator, by that promoter, if it's an operon, right, they will not be expressed if RNA polymerase is not recruited. And so you guys all learned the Lac operon before, and that's what it's all about. It's all about multiple genes being controlled by one region and whether or not RNA polymerase can be recruited or not. That's the basic gist of it. I know it took me about 3 times to really understand the Lac operon. I'm guessing I wasn't alone in that. And hopefully everyone will leave this class understanding it better. Okay, so what did I write here? The interactions between regulatory sequences in the DNA and gene regulatory proteins determine whether a gene will be transcribed or not. Okay? Yes?
Is there any particular reason why those are not
Because it's controlling expression of genes involved in making tryptophan. So I'm not teaching the trip operon right now, so we'll definitely get into it. But for those who have learned the trip, I'm using it really just as a picture of regulatory regions. But so the question was, is there a reason why tryptophan is binding to the repressor? And it's a really nice feedback mechanism that bacteria have, because it's controlling expression of genes involved in tryptophan biosynthesis. If you have a lot of tryptophan in your cell, it's going to be available to bind to the repressor, which allows it to bind to the DNA and repress expression of tryptophan biosynthesis genes. It's kind of beautiful. Bacteria is pretty amazing. There's a few examples of this. Yes?
So is the single promoter for multiple genes only characteristic of prokaryotes?
Is the single promoter for multiple genes only characteristic of prokaryotes? Yes. There's something similar in eukaryotes called locus control regions, which we'll touch on. But yes. Yes. We'll get there we might get there today. Okay, so how are we doing? Everyone understands proteins need to bind to DNA elements to either allow or inhibit transcription. That right there is regulation of gene expression. If you get that, you're halfway there. Okay? Okay, so this is what it looks like in eukaryotic cells. A tad more complex, but quite similar in concept, right? You have, here's your TATA box, right? So let's look up here, okay? So there's this control region for some gene, right? And you're gonna have the promoter, which contains, that actually shows it nicely, showing it's just a small part within the promoter. You'll have, and then you'll have these other regions, these regulatory sequences that could be upstream and downstream and even in the middle of the gene. And they're going to bind to transcription factors. And so you've got general transcription factors that assemble at the promoter and recruit RNA polymerase, but that was promoted by the activity of other proteins that bind to these other regulatory sequences that I call enhancers. They enhance recruitment of RNA polymerase to the promoter. And this is what it ends up looking like, right? So what's this big, they said it, mediator. This is this big purple guy. So you'll have the looping around of the DNA, and that helps to bring everything together and all the communication between the proteins bound to the enhancer regions and helping to assemble the general transcription factors and recruit RNA polymerase and activate transcription. It actually, there's a lot of different circles and squares and blips and blobs on this, but it's basically the same. You need something that's going to allow recruitment of RNA polymerase. Yes?
How do proteins downstream regulate?
Well, so here, it's just like here, right? So here it's showing it looping this way, but there's no reason you can't have looping back around that way, right?
Right, but you wouldn't loop the other way in the direction of synthesis.
So you're forgetting something. Do all of these stay bound once initiation happens? No, right? All of these go away. All of these different proteins go away. This is about setting up all the proteins needed for initiation. Once initiation starts, all the general transcription factors are gone, and a lot of these transcription factors leave, and you can have a nice clear space for RNA polymerase to go. So that was a good comment that you were about to make. I kind of saw it coming. Yeah. Yes?
Why is that DNA structure necessary for transcription?
Why is this DNA structure necessary for transcription to occur? It's not the structure that's necessary, it's the proteins that are binding, because it's all about regulation. You need something to control whether or not these general transcription factors and RNA polymerase are going to be recruited. They can't just assemble on their own. They need other proteins to bind to regulatory regions elsewhere to help recruit them. So, for example, for those of you who are, you're all doing it, the Gene Expression Project, NF-kappa B, I'm sure someone's read about it. That's a huge signaling pathway that results in import of transcription factors into the nucleus, where they then bind to these different regulatory regions and activate expression of different genes. NF-kappa B, you might have seen through all your reading, activates 1 million different genes involved in the immune response. They're all different. So if this this complex was exactly the same for each one, what would differentiate between one gene being turned on under one condition and another gene being turned on under a different condition, right? So it's how the transcription factors assemble with other transcription factors on the DNA that then create this kind of looped out thing that recruit RNA polymerase to the promoter. That's how gene expression is regulated with such a high degree of specificity.
So is it specific enough where if one of those proteins don't bind with upstream regulators, that transcription's not going to occur?
Is it specific enough so that if one of these proteins don't bind to an upstream regulator, transcription will occur? Absolutely. Or if the protein is aberrantly expressed or aberrantly located to the nucleus, something's wrong for some reason, it'll turn on expression of a lot of genes that shouldn't be on. Yes?
After the first few nucleotides are expressed, is there anything that can slow down the rate of polymerization?
Yes. After the first few nucleotides, so after initiation, could anything regulate elongation? Yes, there are mechanisms for, yes. So there are mechanisms for slowing down the speed of elongation, one of them being secondary structures forming within the RNA. I'm sure there's others I can't think off the top of my head. I'll come back to you on that. Yep. Okay, so are people understanding major concepts here? I'm trying not to get too specific yet. I'll get much more specific later. Okay. It's kind of amazing. As someone who's worked on a fair amount of gene expression I don't know, not even. Like, I mostly work on proteins, degrading proteins, but this still applies. Like, I think the understanding of regulation of gene expression is central to understanding anything in cell biology, virology, immunology, anything inside a cell. If you're organismal or if you're physiology, I'm doing a project with Jay Nelson right now. He works on fish and stuff. I don't know. He works on hypoxia in fish. And so I'm doing all the regulation stuff with him on regulation of hypoxia by looking at gene expression when these fish are exposed to hypoxic conditions, as apparently happens in the Chesapeake Bay. I don't understand any of that stuff. I had to learn it all because it's just not my thing. But it's even relevant to that. Okay. Let's move on. So are we good? Is everyone understanding major concepts? Is anyone completely lost? Does anyone want to cry? Does anyone want to jump up and down and celebrate? No. Sometimes there's like serious faces and they want to relax the jaw. Stand up. Okay, fine. You ever notice sometimes you're like so like tightly clenched while you're thinking? Okay. Regulatory sequences in the DNA. So this is just what I said, but another way to think about it. Regulatory sequences in the DNA upstream from the promoter are bound by transcriptional regulators that can activate or repress transcription. These are called operators in bacteria. You can have an activator, which will bind to some upstream regulatory sequence and helps recruit RNA pol to the promoter, or helps RNA pol bind tightly to the promoter. You can have a repressor bind, which blocks RNA pol from binding. So you're off, right? And you can have these regulatory sequences far from the promoter that require, they're actually they're called architectural DNA binding proteins that help in the looping of the DNA. And this allows the regulatory protein to interact with RNA polymerase through mediator. This is very simplistic drawing. Okay. Let's go back to enhancers, right? So enhancers are these distant regulatory regions. They're complex regulatory sequences located often more than 1 kb, so more than 1,000 bases away from either side of the gene, upstream or downstream from the gene, and they are common in eukaryotes. Okay, so it would look like this, right? If it was upstream, it would be over here, promoter, coding sequence. Downstream, you'd have your promoter, your coding sequence, it'd be down here, right? And then you've got this looping out that happens. Okay. Okay. So let's talk about eukaryotic cells and these complex. So I was just answering the question over here before. We're talking about how multiple proteins are involved with binding to these enhancer regions that affect gene expression of certain genes, or almost all genes. And so the beta globin gene is an example of these complex regulatory regions. So you have one regulatory region here, you have another regulatory region down here, and look at this. So here's your Tata box. So that is where your general transcription factors are going to start assembling. But you have all these different proteins that bind and assemble under certain conditions. And this is all because beta globin genes are only expressed in certain cells at certain times during development. So you're going to have this interplay between activators and repressors, and that's what that's showing here. These, the lemon and the banana. I don't know why they drew this fruit. Isn't that weird? It's like they were getting crazy. They're up late at night, and they're like, how should we draw this? Fruit. Let's do fruit. What's this? Is this a zucchini? I don't know what that is. Or what is that? Who's Indian here? Anyone Indian in here? No Indian people in here? That's it. I bet there's... you're in. Yeah, you. Not deep. What's that? It's like a squash that is kind of hairy. No, it's in Indian food a lot. It's like a melon or bitter melon. Bitter melon? No. Sorry. Sorry to point you out. I love different cultures and their food. Okay. Who goes to H Mart? Anyone? Yeah. Go H Mart. Catonsville. Okay. So let's go back to the point. I got distracted by food. Once a graduate student, always a graduate student. When you're really poor and you're hungry, you'll eat anything they put in front of you. I could digress even more, but please stop. I had to do Destination Towson on Saturday. I represented the department, which was really funny. And so I had all these parents and incoming freshmen sitting before me. And I was trying so hard not to get out of control. I almost did. Okay, so all these different fruit regulatory proteins assemble on these enhancer regions, these regulatory regions, and regulate whether or not transcription is going to happen or not. And notice there are these two different, these are repressors. Don't memorize what these specific proteins do, please. Just understand the concept that sometimes you can have a repressor that binds the same site as an activator, and that's a level of regulation. If you upregulate that repressor, remember, repressors are proteins, their transcription is controlled too. If you upregulate that repressor at certain times in development, then you're going to have more repressor, less activator, and you'll still get repression of a gene, right? Okay. So that's two, they're showing two here. So there are many different regulatory proteins, and they act as activators and repressors, and they control beta globin expression in the correct cell type during the correct point of development. So now I have to say to you, okay, so you understand all these proteins are recruited all over the DNA, and they control whether or not gene expression is going to turn on or not. So what's the purpose? So yes, one thing is to recruit RNA polymerase, but there are other things that these proteins are doing. They're recruiting other proteins. What kinds of proteins do you think they're recruiting other than transcription factor related proteins? What kinds of proteins might need to be recruited to DNA for gene expression to happen? Now I'm making you merge information that you may have learned months ago with new information. I see two hands here. Does anyone, I see three, I didn't bring my Ah, I didn't bring my sheet. Habiba. Wait, where's there? Hawa. Habiba. Huh? Activator proteins. What kind of so you've got activators and you've got repressors, and they're going to recruit additional proteins. What kind? How about Hawa? Okay, what kind? I'm going to get to you frequent hand raisers. Okay, who did I see first? You? Chromatin remodeling complexes. Remember, our genome is packaged into chromatin histones, right? And they need to be moved around in order to access genes, in order to access transcriptional regulatory regions, right? So you'll have chromatin remodeling complexes, you'll have histone modifying enzymes, right? So I want you to kind of start to bring it all together. Right. Yes. Would topoisomerase be recruited? Take me on the walk of your life.
Well, just thinking because you're going to have things in superprotein action.
Okay.
So you'll need to cut that in.
So is topoisomerase recruited at this point? I believe. Now, topoisomerase is going to be recruited based on when this. I think once the DNA starts to open. Yep. Okay, we're getting there, guys. How are we doing? Is everyone's mind being blown? No. Is everyone like, why is she going so slow? I think I'm on like slide 5. Okay. Someone said yes. I heard someone say yes. Is someone so bored? Who's bored? Who wants to go home? Okay. Okay. So let's talk about locus control regions. Well, first, that was the beta globin gene we were just looking at. I was saying how it's regulated. So globin genes are only expressed in certain cells, right? What kind of cells? Erythroid. Yeah. Pre-red blood cells. Yep. And there are multiple globin genes, and so there's multiple controls here because you need to have them only on in red blood cells or related erythroid cells, and you need to have certain genes on at certain points of development. Okay, and so what we just looked at before is a lot about when certain genes come on during development, right? Because here's birth. So here's, I guess, conception all the way through 48 weeks. And so you'll have all these different globin. You'll have increased expression of these, and then increased expression of this one here, and then increased expression of this one here. Differential expression of different globin genes at different time frames during development. And so that's what we were talking about there. But then there's this locus control region. Okay, and this is where I was relating it to an operon, but completely different. So don't ever say there are operons in eukaryotic cells, or else someone's going to fire me. But I find it to be similar because an operon is controlled multiple genes by one control region, and this is a locus control region is similar because you have the globin genes clustered together in the DNA, and you can actually regulate the whole bunch of genes based on activation or repression of the locus control region. Yes.
Now, this is an example that globin genes are located in co-parts of one another, but a lot of times associated proteins are located close to.
Right, so then you wouldn't have this. Right, so this only works where you have a related genes clustered together. If they're on different chromosomes or very far away, you're not going to have this. Yeah. So, did I? No. Okay. So, basically, in erythroid cells, this is going to be activated. In all other cells, it'll be repressed because you don't need expression of those genes in your neurons, right? That would be really weird and silly, inefficient. Okay, so this is an example of locus control regions. I always like to introduce it. It's a small thing. I don't think it's a very common mechanism, but it's a nice one. So what's the advantage of controlling the whole cluster of genes at once? Why would you want to, why you just turn each one on and off individually. Got them all ready to go? Okay, well, let's go the opposite direction from that. Well, right, so think about how all these genes, these genes will never be expressed in a T cell, right? So you may as well make sure that this region is repressed so that you never have activation. Why would you ever want them on? So it's an easy way to repress the whole region, right? And so in red blood cells, yes, you want them ready to go, so you're going to have activators recruited to this local control region, and then each individual gene within has its own regulatory region, so you can individually turn each gene on and off within the erythroid cell. Okay? Is everyone good? So how might this region look in skin cells versus erythroid cells? Like this region here. So would a lot of proteins be bound here? A lot of proteins that are activating be bound in skin cells? No, maybe a lot of proteins involved in repression might be bound in skin cells, right? Okay. So patients, we talked about this briefly before in terms of what happens in beta thalassemia with mutations at splice sites, but also in patients with thalassemia, a form of thalassemia, there's a portion of the locus control region is deleted, which basically shuts all the globin genes off. Yes?
So does the locus control region increase how quickly differentiation occurs during?
Do locus control regions affect how quickly differentiation occurs? Okay. Okay. So the only thing is that, so the question is, do locus control regions affect how quickly differentiation occurs? Because you do have differentiation from these kind of stem cells all the way through the red blood cells, and they're expressing different globin genes throughout. The only difference is locus control region is an all or nothing on or off for the whole bunch of genes. So you can think of it as cell type to cell type, not within the differentiation of one cell type. Does that make sense? Okay. I think we're running out of time. Okay, so let's stop here. God, I did horrible today. We'll move faster on Wednesday.